All right, welcome back. Today, we're going to put on our detective hats and step into the lab to solve a real biochemical mystery. We've got a test tube with a totally unknown substance, and our mission? To figure out exactly what it is. So, get ready, because you are the lead detective on this case. Okay, so picture this. You're in the lab, staring at a test tube full of some clear liquid. I mean, it could be anything, right? So, what's your first move? Where do you even begin to unravel its identity from like a million different possibilities? Well, like any good detective, you start with a big, broad question, and then you start narrowing things down. So here's our game plan, our roadmap for this investigation. We're going to start by just confirming if we're even dealing with a carbohydrate at all. Then we'll get into its behavior, check its size and type, and finally, we'll look for its unique chemical fingerprint. By the end of this, you're going to have a whole toolkit for cracking these kinds of cases. First things first, we need to figure out if our unknown substance even belongs to the massive family of molecules we call carbohydrates. We need a test that's going to give us a clear yes or no answer. Something to tell us if we're on the right track. This is where we get our first really big clue. And for that, we've got a go-to tool, Molisch's test. You can think of this as our universal carb detector. It's designed to react with pretty much all of them, from simple sugars like glucose all the way up to complex ones like starch. So if this test comes back positive, we know we're definitely in the right ballpark. Now, the procedure itself is actually pretty simple. You add the Molisch reagent, give it a good mix, and then, and this part is super important, you very carefully pour some concentrated sulfuric acid down the side of the tube so it forms a layer at the bottom. The key here is you absolutely do not shake it. All the action happens right where those two liquids meet. And boom, look at that. That beautiful reddish-violet ring tells us everything. So what's actually happening here? Well, that powerful sulfuric acid is basically ripping the water molecules out of the sugar, turning it into something called furfuryl. That furfuryl then reacts with our reagent to create this amazing colored ring. So, case file update, our unknown is officially a carbohydrate. Okay, so we've confirmed our suspect is a carb. That's huge. Now we can start to build a profile. And one of the most important things we need to know is about its chemical behavior. Is it a reducing sugar or a non-reducing one? Figuring this out is going to narrow down our list of suspects big time. So what in the world is a reducing sugar? Well, in simple terms, it's a sugar that has a special chemically active part, a free aldehyde or ketone group. This little feature makes it kind of generous. It's able to give away electrons to other compounds. And here's the kicker. Almost all monosaccharides, the basic building blocks of carbs, are reducing sugars. Now, to test for this, we've got a couple of classic tools, Fehling's test and Benedict's test. And honestly, while they both work, 9 times out of 10, you're going to reach for Benedict's. Why? Well, its reagent is way more stable, so it lasts longer on the shelf. It also uses a weaker alkali, which is less likely to mess up the sugar we're trying to test. And crucially, it can give us a hint of how much sugar is there, not just if it's there or not. And this is the really cool part about Benedict's test. It's not just a simple yes or no answer. The color of the precipitate it forms actually tells you about the concentration of the sugar. It creates this awesome spectrum, from green for a little bit of sugar to yellow and all the way up to a solid brick red for a high concentration. It's like a chemical traffic light for sugar levels, which is incredibly useful. But wait a minute, Benedict's test isn't perfect. It's great for smaller sugars, but you might be thinking, what if our suspect is one of the big guys, a polysaccharide? They often don't have that reducing property. For these heavyweights, like starch and glycogen, we need to pull a different tool out of our kit, the iodine test. The iodine test is awesome because it's super specific for these big storage polysaccharides. It's all about shape. Think of starch as this big, coiled spring. When you add iodine, the iodine molecules get trapped inside those coils, and that's what gives us that classic deep blue-black color. Glycogen is a bit more branched, so it gives a reddish-brown. And cellulose? It's just a straight chain, so the iodine has nowhere to get trapped, and we see no change at all. All right, quick recap. We know it's a carb, we have an idea if it's a reducing sugar, and we've checked if it's a big polysaccharide. Now let's get even more granular. We need to figure out its size. Is it a single sugar unit, a monosaccharide, or is it 2 units stuck together, a disaccharide? And what about its basic structure? For this, our next tool is Barfoed's test. And this test is literally a race against the clock. Now, both reducing monosaccharides and disaccharides will eventually give a positive result, that red precipitate. But it all comes down to one thing: speed. The monosaccharides are like little speedboats. They're small and react super fast, usually in under 5 minutes. The disaccharides are bulkier, more like clumsy barges, so they take a lot longer, usually more than 10 minutes. Time tells us everything here. Okay, we're peeling back another layer of the onion here. We're getting down to the fundamental architecture of the sugar. At their core, simple sugars are one of two things: an aldose, which has an aldehyde group, or a ketose, which has a ketone group. Think glucose versus fructose. They're built differently, and we need a test that can spot that difference. And for that, we use Seliwanoff's test. Just like before, our stopwatch is our best friend here because this test is all about speed. Here's the deal. Ketoses, like fructose, react way faster under these test conditions. They produce this incredible, bright, cherry red color in under 30 seconds. If you see that happen, bam, you've got a ketose, no doubt about it. It's an incredibly fast and decisive clue. Okay, team, we have collected a mountain of evidence. We know it's a carb, we know its behavior, we have a good idea of its size, and we even know its basic structural type. It's time for the final piece of the puzzle. It's time to get a positive ID. And this is our forensics. The osazone test is the ultimate tool for identification. When you heat reducing sugars with a chemical called phenylhydrazine, something amazing happens. They form these tiny, solid crystals. And the shape of those crystals under a microscope, it's like a fingerprint. It is absolutely unique for different sugars. And this is where the magic happens. We put a slide under the microscope and the final clues reveal themselves. For instance, if you see fine needle-like crystals, that tells you you're probably looking at glucose or fructose. But then you might see these incredible sunflower-shaped formations. That is the unmistakable fingerprint of maltose. This is the visual proof, the smoking gun that lets us name our suspect and officially close the case.
Whew! What an investigation. We went from a total unknown to a positive ID by following the clues. So now you've got a complete set of biochemical tools at your disposal. Let's lay them all out on the table one last time and just review what each one does.
And here it is, your detective's cheat sheet. It's a logical flow, right? You start broad with Molisch to see if it's a carb at all. Then you check its behavior with Benedict's. If that doesn't work, maybe it's a polysaccharide, so you try the iodine test. You use Barfoed's to tell size, Seliwanoff's to tell type, and when you're ready to make the final ID, you go for the osazone test for the fingerprint. It's a powerful system. And look, this isn't just some fun science experiment locked away in a lab. This stuff really, truly matters in the real world. For example, the same principle behind Benedict's test is used in simple screening strips that test for glucose in urine. That's a critical frontline tool for detecting and monitoring diabetes. This basic biochemistry has a massive impact on millions of lives.
And there you have it. We started with a test tube and a question mark. And by applying a logical sequence of tests, our detective's toolkit, we systematically uncovered the identity of our mystery substance. The principles you saw here today are the absolute bedrock of analytical biochemistry. So, now that you've got the tools, the only question left is, what mystery are you going to solve next?